Hello, everyone. Welcome. I am Miguel Martinez Ramos from the National Autonomous University of Mexico. I had the pleasure to moderate this lightning session on biology and management of fishes and herpetofauna. Juan Pablo Narvaez Gomez from the University of Sao Paulo in Brazil will assist us with the technical support for the transmission of this session. We will have the contributions of 9 colleagues from different regions of the planet, each one representing groups of 2 or more persons. I am sure that Halas, Joao Campos, Carol, Ireri Suazo, Javier Alvarado is the presenter now, Grecia Torres, Tom Jemerson, Eric Fayan, Maglin Suavit, Mohamed Yamil, Arnold Jose Logo Carvajal, which will be the speakers, will give us a very interesting talks. I want to remind you that after the talks, we will have a live question and answer session with the speakers. And remember that you can submit your questions for the speakers at any time during the presentation, but please be sure to do so using the Zoom question and answer button, not use the function of Huba. Use Zoom question and answer button. Also, please indicate the name of the speaker to whom the question is directed, okay? Then we will have more or less 30 minutes for this question and answer sessions. So without more to add or to say, please start the talks. Please start the talk, Juan Pablo.
My name is Alazi Maniz. With your permission, I will present our study with the title Holes in the Policy Net, Sustainable Food Production in Artisanal Fishing Communities and Policy Challenges, a Mozambican case study. Mozambique is one of the highest ranking countries in the Global Hunger Index and is projected to become one of the world's most vulnerable countries to resource scarcity and climate-related disasters by 2050. 2/3 of the population already lives in coastal regions. Of this, 20% are in a situation of persistent food insecurity, depending on coastal fishing, which tropical pinnate shrimps is the most valuable resources, exercising a constant pressure on fish stocks. To understand the broader connection between food security, disaster risk reduction, and sustainable fishing is an essential present and future management policy focus. This makes Mozambique an interesting case to study such management policies by assessing their effect upon authors and their reactions. With the objective of reducing the pressure on and ensuring the regeneration of pinnate shrimps in the juvenile stages, fishing management measures have been implemented since the 90s. From those, an enclosure period is in place where to fish in coastal ecosystems such as estuaries and mangroves is prohibited for all fishing sectors, and since 2010, this includes the artisanal sector as well. To ensure this, gears with small mesh sizes are also banned. The drive of our study is to understand how the enclosure period is perceived and affects the fishers and how they address their food and income needs in the artisanal sector. The research design drew upon qualitative and quantitative methods involving literature review, anthropological participation, semi-structured interviews, and user surveys. Different sites were sampled. However, the case study concentrated on Shwabudembe village on the number 1, aiming to understand the life practices of artisanal fishers. Our results show that pinnate shrimp is the most valuable resource in a multi-specific catch in the artisanal fishery, being sold in the nearby markets. And in contrast to what happens in the industrial sector, where most of the bycatch is discarded discarded in the sea due to the reduced commercial value, in the artisanal fishing, the bycatch is used by the local communities as their source of animal protein in their diet. And assessing the knowledge about the law among the interviewed people, we found out that most of the interviewed recognize the need of license, for instance, and also are informed about the implementation of the enclosure period. And also, most of them believe that the government is the institution and the institutions are the ones responsible for the implementation and enforcement of the law. But regarding the use of the nets, and although they have all this information, we could see and crossing the observations and surveys about the nets used, we could see that although during, and we did this for the enclosure period and normal fishing period, we could see that although the numbers have dropped in the enclosure period, they still fished and targeting shrimps in the with the gears that were prohibited by law. And this, with this, we could see that they were targeting all year round species in their juvenile stages. Um, when analyzing the enclosure period as a management policy in place and crossing with the respondent answers about other sources of income, we could see that in the current scenario where the fishers are not engaged in any alternative income generating activity, the current management policies provide a threat to fishing communities' daily food supply and subsistence, as there is no legal incentive for compliance and options during this period. Summarizing our results, we can present the situation of coastal fisheries in Mozambique as a wicked problem, where fishing practices and policies represent a threat to both ecosystem and communities. And having said that, and based on existing solutions and practice, we suggest actions that have the potential to improve policy effectiveness by ensuring alternative income during the enclosure period. We believe that to ensure long-term sustainability, climate smart and proof solution must be assessed due to the strong connection between food security and climate change. For instance, complementing food farming with climate smart cash crops, taking the advantage of existing agriculture practice and culture is an untapped path with a high potential for solution innovation. The sisal is a good example of an underexploited crop with this kind of potential. And however, we believe that an interdisciplinarity approach and interconnection between stakeholders such as the government, other government institutions, fisheries, and NGOs and donors needs to be straightened to ensure this process. Having said that, I would like to thank you for your attention. We are open for questions. You can write to us. Thank you very much for your time for this presentation.
Hi there, my name is João Campos Silva. I am a postdoc at Norwegian University of Life Science and also director of Institute Juruá. I'm happy to be here to show you a positive conservation story from Brazilian Amazonia. My presentation is entitled Local Communities Bring an Amazonian Giant Fish Back from the Brink. Well, freshwater megafauna is composed by iconic species that are very important for local culture, local economy, and local food security. However, declines of wild population has been reported worldwide, and this is especially concerning for large species of fishes. In Amazonia, we can see the same pattern. Many important species were overexploited over the last century, including black caiman, giant otter, manatees, and also the giant arapaima, a very important species in Amazonia. So the arapaima was overfished in the last century, being almost extinct in many localities. However, to reverse this population decline pattern, local communities in partnership with researchers from Instituto Mamirawa developed the co-management of Arapaima. And they created the rules of the co-management, which is quite simple but with very strong results. Basically, local communities build wood houses in the entrance of the floodplains lakes and take turns to protect these lakes, avoiding illegal fishers and poachers. And then the community can exploit a quota of the arapaima that use these lakes. So here, my goal is to show you some preliminary analysis regarding the large-scale outcomes from this promising model. To do it, we analyzed the Arapaima census from 30 management areas, which involves more than 400 floodplains lakes from 2001 to 2021. And we can see a very strong pattern of population recovery. Basically, the wild population of Arapaima has been increasing more than 500% in some areas, and most of these areas has observed an increasing in Arapaima population more than 100%. So this is very interesting in terms of conservation. And beyond the population recovery pattern, we can see many other social benefits, including income generation, infrastructure improvements, gender equity, social security, food security, territorial autonomy, and increasing in the self-esteem of local communities. We used to say that the Arapaima is the fish of change that have been combining um, biodiversity protection and local well-being. So the main principles that can be observed in these management areas is strong leadership, social capital, respect for local rules, well-defined boundaries, participatory monitoring, explicit regulation, adaptive management, subsidies, and local surveillance. It's also important to note that Arapaima has a very strong importance on local culture and also a high abundance and a high economic value. So this is very important to contribute with the local engagement of the project. So the Arapaima is a very important model that can show you how can we integrate people well-being and protection of biodiversity. Thank you very much. If you would like to know a little bit more about this model, just let me know. Thank you.
Hello, I'm going to talk about how feeding trades could shape the spatial distribution of the fish family Serrasalmia in the Amazon drainage basin. This is part of my dissertation project at Mississippi State University in collaboration with other institutions. So for the introduction, we have that the Amazonic region is characterized by a high diversity in different biological groups. Regarding fishes, the Amazon drainage basin has around 50% of the world's freshwater fish diversity. Questions about the causes of this high diversity are a recurrent topic for ecologists and biologists. So the presence of different aquatic habitat types can promote that diversity through food resources, for example, and this is a factor that is related with the species distribution. The Serrasalmia family is a fish family that is a great model to test hypotheses related with how feeding traits affect species distribution, because this family, that is the family of the piranhas and pacus, has a diet gradient from frugivory to piscivory, as you can see here in this phylogenetic tree, where each color represents a feeding group. Also, this family is mainly distributed in the Amazon drainage basin, with at least 73% of the species in this region. For the methods, we used Serra Salmide occurrences from the Amazon Fish Project and satellite-derived products as floodplain delineation and land cover rasters to build two variables as proxies of habitat use. These variables are floodplain extent, that is the floodplain area around the occurrences in 3 different scales, and the other one is habitat heterogeneity, that is a Shannon diversity index using the land cover types at 3 different scales. For the feeding yields, we use diet data that was retrieved from literature review of stomach content, and we applied a quartile classification system considering the proportions of the food items in the stomach content database. For analysis, we use phylogenetic generalized least square to test for the association between the habitat use proxies and the feeding yield. We use this analysis as this allows us to account for phylogenetic relatedness among species. We also use estimated marginal means to make pairwise comparisons between the feeding yields. So for the results, we have that regarding our habitat use proxies, there are no prominent differences visible on these graphs, but for both variables, frugivores have a higher mean and herbivores have a slightly lower mean than at the 5 kilometers scale. This pattern is similar for the other scales used in this study. Based on the results of the phylogenetic generalized least squares, we found that frugivores are associated with higher areas of floodplain and that this is different from herbivores and piscivores. Regarding habitat heterogeneity, frugivores are related with more diverse habitats than herbivores at the 25 kilometer scale. It means the bigger scale. So for the conclusions, we have that for floodplain, frugivores occupy bigger areas than herbivores and piscivores, and this makes sense as the species in this feeding yield are reported to use floodplain habitats as their main source of food resources. Also, considering habitat heterogeneity, all species use diverse habitats. However, frugivores use more diverse areas than herbivores at the scale of 25 kilometers. And finally, in conclusion, we can say that the feeding traits or diet preferences does influence habitat use among Amazonian fishes. Finally, I want to thank Celine Hezekiel from Evolution and Biodiversity Lab in France and the funding sources at Mississippi State University.
Response of amphibians and reptiles to secondary succession in a tropical dry forest of Mexico, presented by Irelis Suazo, Javier Alvarado, and Jorge Marroquin. Secondary forests are in many regions replacing native tropical forests, one of the most threatened forest systems in the world. Secondary forests might play an important role in the conservation of tropical biodiversity. It is important to determine the potential of these secondary forests to support old-growth forest species, particularly threatened animal groups such as reptiles and amphibians. This study was conducted in the Chamela region and the Chamela-Cuixmala Biosphere Reserve, both located in the western coast of Jalisco, Mexico. Our study area is part of the Mexican Pacific lowlands, which is a hotspot of herpetofaunal species richness and endemism, supporting about 1/3 of the species of amphibious reptiles known in Mexico. To compare the structure and functional diversity of herpetofauna communities among suggestional stages, as well as the response of these communities to the cumulative effects of hurricanes, we conducted a long-term survey from 2009 to 2018 of frogs, lizards, and snakes within the successional stages. We selected a total of 15 1-hectare plots representing the following 5 successional vegetation stages: pasture, early forest, young forest, intermediate forest, and old-growth forest. There were 3 replicates for each vegetation successional stage. We registered about 4,800 individuals of 62 species of frogs and reptiles. In general, there was no linear correlation between animal abundance, diversity, and richness and successional stage for any faunal group. We observed lack of habitat specialization correlated with the existence of relatively low levels of beta diversity among forest successional stages, particularly when compared with beta diversity within stages. However, we found marked changes in estimated richness and composition of frogs, lizards, and snakes among successional stages in response to Hurricane Hova. Modification in vegetation structure as a result of hurricane paths promoted particular changes in each successional stage and taxonomic group. Secondary forests at different stages of succession may attenuate the negative effects of an intense, short duration, and low frequency natural disturbance, such as Hurricane Hova, on successional herpetofaunal trajectories and species turnover. The cumulative effects of hurricanes might cause dramatic changes in the structure of biotic assemblages within hours of landing. Our results showed that accumulative effects of hurricanes, a low intensity and a high intensity hurricane, resulted in a homogenization tendency among successional stages, suggesting a negative effect for ecosystem functioning. We observed that under the impact of low-intensity hurricanes, successional stages might function as buffers that promote herpetofauna resilience. However, cumulative effects of hurricanes resulted in a homogenization tendency among secondary successional stages, suggesting a negative effects for ecosystem functioning. The role of successional stages as promoters of the resilience of amphibians and reptiles is modulated by the type and intensity of disturbances.
Hola a todos, les voy a presentar el estudio de anfibios y reptiles y su implicancia en la gestión de la Reserva de Biosfera Huascarán en Perú. Yo soy Grecia Torres, tesista de pregrado de TID y egresada de la carrera de biología. ¿Cuál es el objetivo de este estudio? El objetivo general es evaluar la biodiversidad y la asociación con su entorno en las comunidades de anfibios y reptiles de la Reserva de Biosfera Huascarán, a lo largo de una gradiente altitudinal que abarca los 2.500 metros hasta los 4.700 metros sobre el nivel del mar. ¿Dónde está ubicada nuestra área de estudio? Nuestra área de estudio se encuentra en Perú, en el departamento de Ancash, localizado en la Reserva de Biosfera Huascarán, cuya zona de núcleo está categorizado como parque, el Parque Nacional Huascarán. Nuestras áreas de estudio se encuentran hacia el lado este y oeste de la Cordillera Blanca, que es esta zona en blanco. Hacia el lado oeste se encuentra el Callejón de Conchucos, cuyos valles abarcados son Cajavilca, Puruay, Vesubio y Huichilanga. Y para el lado del Callejón de Huayla se encuentra el valle de Yanganuco, Coju, Rejucolta y Rúbrica. La metodología utilizada: se delimitaron unidades muestrales con una distancia de 200 metros a lo largo del gradiente altitudinal, y la evaluación utilizó la técnica de relevamientos por encuentros visuales para la búsqueda tanto de anfibios en ambientes acuáticos, cuerpos de agua, y en zonas terrestres para reptiles mayormente. ¿Cuáles son estos resultados preliminares que tenemos hasta el momento? En el caso de anfibios, la riqueza de ellos fue máxima en zonas de elevaciones intermedias y siempre está asociada a zonas con cuerpos de agua cercanos. Las especies reportadas hasta el momento es Pleuroderma marmoratum, gastroteca peruana, que es esta especie que se visualiza aquí, otra gastroteca que no está en este recuadro, y un Phrynops, que es esta especie de aquí. Asimismo, también se ha verificado varios anfibios en estadios metamórficos, como esta gastroteca de aquí. En el caso de reptiles, la riqueza se vio en decrecimiento a lo largo del gradiente altitudinal, y ello estaría asociado a que la temperatura disminuye conforme es mayor la elevación. Pero hay casos atípicos. Por ejemplo, se han encontrado especies como este Leolaemus chavín de poblaciones que están alrededor de los 4.500 metros y se encuentra una población de muchos individuos en esa zona, cerca a una zona glaciar. Asimismo, también está reportado el Stenocercus chrysopidus, la especie que se ve acá, que está termolando en esta roquita, y se encuentra el Stenocercus sp1, que se visualiza aquí, y otro Stenocercus que no está en estas figuras. Otro de los resultados preliminares es que en zonas que tienen precedentes de minería no se ha visualizado ni detectado ni anfibios ni reptiles, como es el caso de Cajavilca, que en el fondo de valle, donde se ve la flechita, hay presencia activa de minería. Y en el caso de Vesubio, que hace muchos años hubo presencia de minería. Otra de las cuestiones que más resaltaron fue el hecho de que en zonas con presencia de abundancia de ganadería y coexistencia de reptiles, se ha reportado reptiles con presencia de ácaros. Como se puede visualizar en este punto anaranjado, hay un cúmulo de ácaros invadiendo el individuo. Entre las conclusiones más relevantes se encuentra el hecho de que Leolaemus chavín está en una zona en la cual no estaba reportada según la Unión Internacional para la Conservación de la Naturaleza. Solamente reporta para esta área sombreada de blanco. Sin embargo, nosotros hemos reportado una población grande de individuos, incluso a un rango de elevación mayor conocido hasta el momento. Otra de las cuestiones que también se ha resaltado en este estudio es que tanto anfibios como reptiles están distribuidos en diferentes elevaciones dentro de la zona núcleo de la Reserva de Biosfera Huascarán. Asimismo, también nos ayuda como indicadores del grado de impacto humano en esta zona núcleo, específicamente para el caso de minería artesanal. Este estudio presentó un financiamiento del Banco Mundial, administrada por Concitec y Fondesit, y es gracias también al apoyo de TID y de Cernam Perú, netamente el Parque Nacional Huascarán.
Hello everyone, I'm Tom Jameson, a second-year PhD student at the Department of Zoology in the University of Cambridge. Today I'd like to talk to you about the cold-blooded side of restorative ecology and how we can apply rewilding theory to the conservation of tropical reptiles. In recent years, the rewilding movement has gained particular traction both in conservation practice and the public imagination. This movement focuses on the restoration of natural processes to promote ecosystems that maintain biodiversity and ecosystem function without ongoing human management. One potential rewilding approach is trophic rewilding. This involves reintroducing species so associated trophic cascades can be restored, promoting self-regulating and biodiverse ecosystems. Such approaches have been applied in the temperate northern hemisphere with a focus on the top-down effects of large mammalian predators. Following the perceived success of such schemes in other parts of the world, trophic rewilding schemes are beginning to appear in tropical regions. Tropical regions contain taxonomic groups and ecological guilds absent from the temperate northern hemisphere. To be successful, rewilding schemes must also consider these groups. For example, most tropical ecosystems contain large predatory reptiles that have strong top-down effects on ecosystems. They're therefore also key candidates for rewilding projects. In our current project, we use one group of large tropical reptiles, the monitor lizards or varanids, as a test case for the wider application of trophic rewilding theory to large reptiles. If we wish to restore functioning ecosystems and communities, it's essential that we know what such a community actually looks like. Data from the modern world and recent history is pretty limited. It's heavily biased by thousands of years of anthropogenic extinctions and extirpations. We must therefore look further into the past to identify healthy, functioning ecosystems and communities. This is exactly what we're doing with varanids, constructing baselines of community structure for the Holocene and Pleistocene using a combination of machine learning-based models and the fossil record. In order to model varanid communities of the past, we first need to identify what environmental covariates to use as inputs for our models. To do this, we investigated what environmental covariates are most important in determining the distribution of modern varanid species. Here we built species distribution models for extant varanid species, incorporating a wide range of different environmental covariates. We measured how important each covariate was in determining the distribution across multiple varanid species. The plot shows each of the environmental covariates we tested here on the x-axis. Now here we color code them by category. The y-axis shows the overall importance and ranking of each covariate for all varanid species. Climate covariates in red on the left of the plot have overwhelmingly high importance scores when compared to other covariates. As such, we can be confident that climate-based models can capture the majority of variability that we're interested in. We can therefore use climate data to reconstruct varanid communities through time. Robust paleoclimatic reconstructions exist for much of the Holocene and Pleistocene. Now, these are the key periods from which rewilding baselines are typically drawn. We therefore use this data as the input for our models. Here, we're not directly interested in the distribution of individual species. Instead, we're interested in their ecological role. A good proxy for ecological role in varanids is body size. Conveniently, reptile body sizes are typically correlated with climatic factors. We can therefore use this relationship as the basis of our modeling. So we use machine learning here. We train models on the modern distribution of varanid body sizes and climate, and then use these models to map the distribution of varanid body sizes in the past using paleoclimatic data as the input. In the example reconstruction shown on the right for 120,000 years ago, we can see that based on climate data alone, we would expect mean varanid body size to be much larger than it is today across much of the world. Here we see that it's greater than 900 millimeter snout vent length throughout most of Asia, Africa, and Australia. Using this approach, we've developed models of varanid size distribution for the Holocene and late Pleistocene with a resolution of 1,000 years. Next steps in this process will involve integrating the fossil record into our reconstructions to further improve the accuracy of our baselines. In conclusion, we develop a method for building restorative baselines for reptiles utilizing machine learning methodology. We apply this methodology to reconstruct baselines throughout the Holocene and Pleistocene for varanids. This data can be used to identify ecological gaps in modern communities and hence targets for rewilding projects. That's all from me. Thank you very much for listening.
Good day, everyone. We are thankful to ATBC for giving us the privilege to present and share our research in this platform. Philippines is one of the megadiverse countries in the world that holds high concentration of terrestrial endemic fauna per unit land area. This diversification is intrinsically linked to the complex geological history of the Philippines, forming 5 biogeographic regions and diverse habitats. Earlier study shows that Philippine karst ecosystem houses unique and specialized form of life, particularly on amphibians and reptiles. However, just like any other habitats, this habitat is threatened with different anthropogenic pressures. These as in our study entitled Amphibians and Reptiles in Limestone Karst Habitat on the Nagat Islands, Distribution, Status, and Implication for an Island-Wide Conservation. So what we did is we identified the different sampling location and surveyed those sites through the permit granted to us by the Department of Environment and Natural Resources. So survey was done by establishing 48 transects with 100 by 10 meter, combined with visual encounter survey, microhabitat search, and different trapping methods. With this, we identified 16 sampling sites concentrated in 8 localities within 4 municipalities on Western Dinagat with limestone karst habitat. Identification was done through the published accounts of amphibians and reptiles in the Philippines and preserved using proper preservation protocol. This unraveled 58 species of amphibians and reptiles in 5 hectares surveyed karst ecosystem on Western Dinagat, documenting 39 endemic species and 19 non-endemic species. This includes 13 species of frogs in 6 families, with 1 threatened species, Platymantis shruburi, 26 species of lizard within 4 families, with 1 threatened species, Hydrosaurus postulatus, 16 species of snakes within 5 different families, 1 turtle and 1 crocodile, with 2 threatened species, Ophiophagus hana and Cora ambonensis. Most of the species we recorded are distributed in Mindanao biogeographic region, together with species distributed in oceanic Philippines, including and Dinagat Island endemic species and the globally widespread alien invasive species. Historically, DuPont and Rabor in 1973, who studied birds of Dinagat and Siargao, facilitated the first herpetological research on Dinagat Islands and collected herp specimens. 17 years later, a collected specimen of DuPont and Rabor was formally published by Ross and Lazo on the Philippine Journal of Science in 1990. After 26 years, Sanguila et al. Published a new herpetile accounts focuses still on the northern part of Dinagat and added 23 new species records. In this study, we focuses on Western Dinagat and accounted 17 new island records, summarizing the 95 species of amphibians and reptiles in the whole Dinagat Island group. So this study highlights the distribution of 17 species with new island record, of which 4 are potentially new species, which belongs on the family Ceratobatrachidae genus Platymantis, and 1 from family Geconidae genus Certodactylus. However, with no legislation, those species are threatened with different anthropogenic pressures. In conclusion, karst system on Western Dinagat is a home to diverse, endemic, and specialized amphibians and reptiles. This study unravel additional species distribution and potential new species discovery on Dinagat Island group. Significantly provides a baseline information and useful in advancing conservation initiatives on karst habitats on Dinagat Islands. This study would not be possible without the following institution and the help of the local community who joyfully assist us. So that would be all. I am Earl Fayanti Maglangit from the Philippines. Daghang salamat till we set sail again on Dinagat Islands.
Hi, welcome everyone to this Lady Talk. My name is Shuaib Mohammed Jami from the University of Lagos, Nigeria. I'll be presenting on the histopathological alteration in some tissues of Enteroclerus exposed to diclofenac with amniotic potential of neem leaf as a duration indica. The quest for quality life expectancy has increased the use of pharmaceuticals, both medical and veterinary importance, and as thus there's increment in the release of untreated and poorly-treated sewage from pharmaceutical industry into water bodies around the world. And of all pharmaceutical drugs encountered in water bodies, non-steroidal anti-inflammatory drugs, as commonly called NSAIDs, has been reported to be found in water bodies, and due to the numbers of these drugs which are found on the global market, about 50 and more, including the diclofenac, ibuprofen, naproxen, among others. Have they report shown that these drugs are capable of inducing lesion and are capable of being a toxicant in aquatic organisms, particularly with diclofenac. However, little to no attention is paid to the adverse effects of these drugs on living organisms, particularly aquatic biota in the study area and other regions of the world. Moreover, laboratory studies on the use of medicinal plants as a potential amniotic agent on the toxicological impact of pharmaceuticals on fishes are scanty. As a result, these present studies aim to assess the histopathological lesion induced by lead and sublead concentration of diclofenac in the whales, liver, and kidney of juvenile catfish Enteroclerus with an amniotic potential of neem leaf. The juvenile of Arctic catfish used in this study was procured from a fish class in Nigeria, after which the glycophenol solution, which is the toxicant in this case, was prepared using 1 gram to 100 ml of 1 gram of glycophenol to 100 ml of distilled water. After a successful age finding test, the fishes were exposed to acute exposure with varying concentration, including 4 milligram, which is the lowest, and the highest, which is 20 milligram per liter, to determine the 19 hours LC50 of the toxicant. Subsequently, the LC50 was obtained using the carbon arithmetic method, and the value is 10.8 milligram, which is also used in the sublethal concentration for 30 days. The neem leaf, which is as a director indicator, was obtained from the surrounding areas of the University of London, and it was identified in the Department of Plant Biology of Faculty of Life Sciences in the same university, and prepared according to Katisal et al. In 2019, 2008, which is those included in the fish diet, respectively. At the end of the exposure period, the vital organ of the fishes, including the liver and the kidney, were obtained from the fish and preserved in 10% formalin bottle, which is then transported to the laboratory for analysis. Now, showing on the screen is the photomicrograph of the gills of the fish of the Herpetofauna exposed to diclofenac for 96 hours, and the other one is for 30 days. Several lesions were observed in the gills from mild to severe, including the lamellar vision, epithelial lifting, rupture of epithelial cell, hemorrhage, and among others. Whilst the use of 1%, the inclusion of 1% neem leaf, as shown here, able to ameliorate the normal histology of the fish, of the gills. And also here is also the liver, the photomicrograph of the liver of Herpetofauna exposed to 90 hours and 30 days respectively. We have several the major histopathological lesions observed in the liver include the evacuation, nuclear alteration, fatty degeneration, and necrosis. And similar cases as observed in the gills, 1% inclusion of neem leaf able to ameliorate the normal histology of the liver from others. With increase in use of neem leaf, that is 2%, and 4% turns out to be a potential toxicant. And lastly, this is the photomicrograph of the kidney exposed to 90 hours and 30 days respectively. We also have several lesions observed in the kidney, including disorganization of the cells, hypotrophy, and necrosis, among others. And the use of 1% neem leaf also able to ameliorate the liver, the kidney tissue, the kidney cell from the toxicant, that is diclofenac. An increase in the use of the neem leaf, which is the ablative agent, turns out to be a potential toxicant. This is observed in the photomicrograph as shown on the screen here. And lastly, the study showed that the diclofenac is a potential toxicant on fish with 1% inclusion of neem leaf able to ameliorate the normal histology of the tissue. This may, however, result in commercialization of the plant for large scale.
Hello everyone, I am Arnold Lugo Carvajal. Today I am presenting our research entitled Fish on Fire, Cascading Effects of Floodplain Forest Fires on Amazonian Fish Communities. That is part of my PhD thesis about the resilience of Amazon floodplain forests. My supervisors are Drs. Milena Holmgren, Peter van der Sleen, and Jansen Zwana. Now let's move on to the content. Climate change in the Amazon has increased the frequency and severity of extreme events like severe droughts. Those events facilitate burning all kinds of forests, including floodplain forests, that are important ecosystems for aquatic organisms like fish communities. Past studies performed by Flores found strong experimental evidence of a possible salinization process happening on the burned Amazon blackwater floodplains. Flores found that blackwater floodplain forests have a low resilience to fires, regenerating very slowly after one forest fire event. Flores found also that after repeated fires, those environments can remain in an open vegetation state for several decades after burning, which favors soil erosion and facilitates their transition towards a white sand savanna-like vegetation as an alternative stable state. Considering the ecological importance of fish-tree interactions for the floodplains, then emerged our main research question. What are the effects of forest fires on fish communities and the feedback on the forest recovery? And our specific question presented today is how do fires change the abundance, diversity, and composition of fish communities? For answering that question, we sampled in total 27 sites in the middle Rio Negro at the central Amazon. Those 27 sites were divided into 3 habitat types: 9 unborn forest sites, 9 fire scars that were previously identified and measured analyzing Landsat and Google Earth images, and 9 white sand savanna sites. In those places, we sampled fish during 2 consecutive high water seasons, using 9 gills per site during 24 hours. What do we have found? Well, regarding the fish composition, here I am presenting a relative abundance direct ordination graph, where we have the fish species list at the right side column, and the relative abundance distributed in the 3 columns from the left, representing the 3 studied habitats. Thicker lines represent larger relative abundance. We found most of the fish species in the unborn forest. We found less species in the fire scars, and even less species in the white sand savannas. We also found a group of fish species present in all 3 environments, almost equally distributed among them. So we found difference on the fish community composition among the 3 habitat types, where fire scars showed an apparently intermediary composition between onboard igapos and white sand savannas. This pattern was very similar to the vegetation shift pattern found by flores after repeated forest fires in igapos, where fire scars showed an intermediary floristic composition between pristine igapos and white sand savannas during a possible savanization process. What are the reasons for the composition and relative abundance difference? Well, one of the possible reasons is the food offer difference among the 3 habitats. For verifying this, we classified the fish species by functional trophic groups based on previous reports. We found a tendency of decreasing on the omnivory from the unborn forest towards the white sand savanna, and an increasing in the detritivory in the same direction. We also found a significant reduction in the total fish biomass and in the mean fish size as fire scars become larger. Overall, our results indicate that fires trigger profound shifts in the fish assemblages of blackwater floodplain forests of Rio Negro, from highly diverse assemblages in unborn floodplain forests towards a depauperate condition, characterized by a predominance of smaller-sized species in big fire scar sites, and that effect are proportional to the fire scar size. These results are relevant, we think, because flooded forest loss by fire is expected to accumulate as the frequency and severity of extreme drought events increase. Thank you very much for your attention.
Okay, thank you very much for all these wonderful talks. Now we will have a session of questions and answers. And the first one is for, let's see, for Joao. And it's come from Roxana Patricia Arauco. And she asked that Arapaima is a genera well known to recover fast and actually is known as well to be an invasive species to the south rivers of the Amazon. So this recovery you mentioned was secured basically by protecting their natural riverways. Is the growth rate one similar to those before the drought, or are they even higher? Is there a way to know?
Yes, thank you, Roxana. It's a great question. Yes, Arapaima has become a concerning invasive species in many parts of the world, actually. But in this place that I show the results, it represents like the natural home range of Arapaima. So we are talking about the natural population recovery, which is result from the local protection. Basically, local communities are protecting the floodplains, and the Arapaima is recovering. It's a little bit hard to know the past population size of the Arapaima. So it's hard to know the carry capacity of each lakes, but I think that this is a good strategy to evaluate the carry capacity of those lakes, which could be similar of the past population size, but it was still never done.
Okay, thank you. And next question is also from Rosanna Patricia Arauco to Tom Jameson. Could you please explain a little bit what is ecological gap? What you mean for the ecological gap? You mean niches or that has not been explored? And why it is relevant to dragons?
Yeah, sure. So thank you for the question. So we're talking about an ecological gap. We're talking about potentially an empty niche. So say if we reconstruct our community in a certain area for 2,000 years ago, and we say, okay, 2,000 years ago, we can say that there were species of this size range. So say we had a bunch of big species and a couple of small species. And if we look now, there's only just the small species, then we'd say, okay, there's a gap there in that community. We're missing the big species, we're missing their ecological role. And that might be somewhere where we'd want to consider some restorative ecology actions there to actually fill those niches and restore that community.
Okay, thank you. And Jesper Pedersen asked to Hales, I understand that the enclosure period is important to protect the marine system. How could these policies be improved to benefit the artisanal fishing communities? For example, how could you sell the idea of the sisal, the fiber crop, to authorities, policymakers? And could the industrial fishing sector finance the implementation of the agricultural practices?
Thank you. This is a very interesting question and very difficult to answer because I don't think there is one specific answer to solve the whole problem. But I think as the question already comes with some options, I think for instance, the how can I say, dividing the question into parts. I think this part, the thing about the policy itself, the way I see is that the way to make sure that the small scales apply the management measure is to find options that make them have alternatives during this period, because I think there was I have to explain that this management measure comes and they are not allowed to fish now during 5 months, which means during this time they need to have alternatives of income and food, and they don't have it according to our study. And we need to find, and I think policymakers need to find incentives and work with them for them to have options of what to do during this time. And when we talk about, for instance, the sisal as a crop that can answer to this gap, is because in our survey, we also asked the fishermen what was the other activities that they were engaged in. And they are all engaged in agriculture as their second activity, but they don't see agriculture as an option of surviving or an option that brings them income. But it can be a very useful activity because it's already an existing practice that can be, how can I say it in English, can be capitalized. We can use this as a because it's difficult if you bring different and new things that the local communities are not used to it, because they feel like it's very new, they don't have time to learn or to embrace or be part of. But if we bring
a solution that will take potentialize activities that they already do, such as agriculture. I see like something that can make it easy to bring this transition. And sisal crop is a crop that exists in Mozambique, and differently of what happens in Kenya and Tanzania, that is already an industry. In Mozambique, it's not yet an industry and can be a potential industry for small-scale fishermen, I think. I hope I asked the question. Thank you.
Thank you very much for your question, answer. And I have a question for Carol. And what's very interesting to learn about these feeding traits and the species distribution in this family of fishes. And I'd like to know whether you have some idea if species more closely related, phylogenetically speaking, and also more similar in feeding traits are more niche differentiated than the species that are more different in phylogeny and traits.
Okay, thank you for the question. Well, actually, I don't know. These are preliminary results of my project, but we use phylogenetic generalized least square to account for that relatedness within the family. So actually, we were trying to consider that relationships in the residuals of the model. So we can be sure that the associations were between the species and the habitat use were because of the species and not the phylogenetic relatedness among them. So we haven't tried that. We haven't asked ourselves that question yet, actually.
Yeah, it's very interesting also to know whether the species that have more narrow distribution have different feeding traits and phylogeny relations with those that are more wide distributed, no, in your region.
Yeah, I mean, I have regarding like distribution and actually aggregation, I tried to do like some analysis about aggregation, but for fishes like freshwater fishes, it seems like it's not that easy because of the constraints of the rivers. I mean, the structure of the habitats. So my study, it's like the entire Amazon basin scale. So I have seen some aggregation and studies in small areas, but when you're talking about the entire Amazon basin, the things are a little bit different. So yeah.
Yeah, okay. That I comment this because linking the previous questions regarding the management policy and so on, the basic biology of the species are very useful to identify some practices and also policies and management, no, that can be important to take into account, no, in order to make a sustainable management and conservation of the species. Okay.
Yeah.
Thank you.
Of course, thank you.
And for Grecia, I want to ask a question in Spanish and we'll translate. Quisiera preguntar, Grecia, si tú esperas que pueden existir cambios diferentes o diferenciales a lo largo del rango altitudinal que estudiaste debido a los cambios climáticos que estamos observando a nivel global, ¿no? Sobre los grupos de anfibios y reptiles que tú estudiaste.
Sí, en efecto.
Let me to translate. No, the question is if she expects differential changes in the elevational range of the amphibians and reptiles she studied due to the global climate change. Okay.
Okay. En realidad, lo que se está verificando en esta área de estudio es que
efectivamente sí hay una variación, ¿no? Conforme se va viendo, es muy diferente el cambio entre anfibios y reptiles porque los rangos elevacionales más afectan en cuanto a la temperatura para reptiles, pero en anfibios no se está verificando que muchas zonas de estas áreas están siendo ocupadas por anfibios, ¿no? Sin embargo, con los cambios de de características como temperatura, humedad, es definitivo que estos individuos o estos grupos taxonómicos van a buscar estos sitios más adecuados, ¿no?
Bueno,
Entonces, yo considero que en cambio climático sí va a variar mucho el hecho de qué lugares van a ocupar estos grupos taxonómicos.
okay, the response is because the reptiles and amphibians have a different physiologies regarding to temperature, for example. She can expect that the influence of climate change will be different for reptiles and amphibians. And reptiles tend to increase with the temperature, no? And then these species are expected to climb to higher elevations, while amphibians depend more on humidity. And the humidity could be more widespread along the elevation gradient. Then these changes could be observed in the future. Okay, thank you. Gracias. Then there are questions for Tom. The reintroduction of predators can increase biodiversity is your message, no? But I wonder whether the trend of herbivory increase, why the herbivory increases and reduce predators, and there is a consequence on biodiversity, no? Declined biodiversity. But how general is this observation? Do you have more studies that can be detecting this trend or this pattern?
Yes, so I mean, I guess it's the general trend of the trophic cascades we get with removal of top predators. It's a pretty sort of widely studied phenomenon around the world, where you, as we mentioned, you remove your top predators, often leads to massive increases in herbivore populations. As a result, you get overgrazing, degradation, loss of ecosystem services and biodiversity. And so a lot of the classic studies, as sort of I mentioned, a lot of this classic work has been done in the temperate northern hemisphere, so particularly the loss of wolves from Europe and North America. We also see similar studies coming out of sort of subtropical regions and tropical regions in Australia, loss of top predators there, and massive increases in, this is typically macropod populations, so kangaroos, leading to massive overgrazing and all sorts of ecological problems as well. So it's a fairly sort of, I think, universal pattern. I think there's quite good evidence for that. But I think, as in many areas of ecology, it's a pattern that is better understood in the temperate northern hemisphere than it is in the tropics, for basically the reason that there's been less investment in tropical research. Often it's been harder to do. And it's an area that certainly would benefit from more study. It's no coincidence that the sort of tropical and subtropical regions we do have evidence for in are places like Australia that have this sort of big colonial and Western history. So there's been a lot of more investment in research there. So it's certainly a field that could do with a lot more investment. Actually people looking into Southeast Asia, Central African tropical ecosystems, where what are the effects of having lost a lot of tigers from Southeast Asia? What are the effects of having lost gharials from most of India? What effects have that had on river systems? So it's actually a really open question in tropical regions, and people should certainly be looking at it.
Yeah, because I can expect that there are some variation in this trend, on this pattern. For example, depending on the network of the species links, in the natural structure of this network. For example, if the frequency or general species change in some ecosystem regarding to other ecosystem, or the frequency or species specialist links, the structure of this network maybe produce some variation in this general trend. No?
Yeah, I think we've got, especially in tropical regions, you have a really interesting issue of redundancy. So say if we go to North America and Europe, that if you have your top mammalian predators, that's maybe just your wolves and bears. Whereas if you go into a tropical region, say you go into the savannas of Africa, then you've got lions, you've got hunting dogs, you've got hyenas. You've got a lot more redundancy in the system there, which is potentially going to have different effects on how removal and reintroduction of things is affecting ecosystems.
Very interesting. Thank you. Thank you. For Ir, Fian, I have a question also. And I wanted to know what characteristics have the species in the island that make them more vulnerable or more prone to anthropogenic disturbances and prone to local extinctions. Do you have some ideas of that?
Okay. Most of the, do you hear me?
Yes.
Okay. Most of the species that we've been working in the Philippines is highly specialized. They come from different habitats and microhabitats. So in this case, we study karst ecosystem in Dinagat Islands. It is the almost the eastern part of the northeastern part of the island of Mindanao. And I think based on your question, I think that based on based on what we encounter on the field during our survey is that most of the cases that this happen is primarily linked to habitat modification, in which more of the limestone or karst ecosystem in the Philippines are mostly quarried, or sometimes gone. Most of the species are not even described or gone to extension because of quarrying. Yes.
Okay, thank you very much. Very interesting. It's very interesting to know this diversity in the islands in Philippines. Thank you.
Thank you so much. Thank you so much.
Okay, Suavi, I don't know much about your field, no, but I wanted to ask you, what chemical or other properties have the neem leaves, which can this function that is amelioration of the liver damage. Do you say something about that? Mohammed? Maybe it's closing your microphone. Or it's not here more. Okay, I will pass to the next question. It's from Jesper Pedersen. And let me say, it's for Joao.
Sorry to interrupt you, Miguel. We are over time now.
It's over time. Okay. Okay. Thank you very much for your participation. This wonderful session. And I think that we learned very much about these important animals for the tropics, and hope to see you soon in any place around the world. Take care. And thank you very much for your participation.